Hey everybody, this is our revision session covering the whole of the genetics topic that we've been learning about for the past term. So remember, genetics basically is about finding out how do our cells control themselves and how do our cells work together to make our bodies work, and how do we as animals and other organisms reproduce to continue our species. So it's basically explaining how do organisms work. Alright, so the first place we're starting is back at a cell, at the cellular level. And remember, the key place we're focusing on for the topic of genetics is down here in the nucleus. So inside the nucleus, that's what holds the DNA, which is a molecule. Always remember, it's just a molecule. It's a really, really long, string-like molecule, but it's still made of atoms just like any other molecule is. Okay. So the next thing is, what is the actual structure of DNA and what does that molecule look like? So we should all remember that DNA has a double helix shape. And a double helix just means it's two half molecules that are spiraling around each other in a helix shape. And if you untwist that helix, it looks like a ladder, basically. So you can think of the DNA molecule as like a twisted ladder. And that means it's quite simple to explain the structure of DNA. Because if you unwind it, just like a ladder, there are two vertical parts of the molecule along the outside, and we call that the backbone. And then across where the rungs of the ladder are, we call these base pairs, because they're two separate parts of the molecule that have a different structure to each other. And there are 4 different bases in DNA, and we call these the nitrogenous bases, just because they have nitrogen in the molecule. And we need to know the names of these because they make up the genetic code. So the 4 nitrogenous bases that you can find in a DNA molecule are adenine, thymine, guanine, and cytosine. And we can just give them their capital letter for short. A and T, G and C. So they make up the genetic code. They're like the letters of the alphabet for genes and DNA. So they've only got 4 letters in the genetic alphabet, basically. Alright, and you can see here in the DNA molecule, the base adenine always pairs with the base thymine. So A always pairs with T, and G always pairs with C. A and T, G and C. That's just the way the molecules fit together, and you'll learn more about that in senior school biology, in VCE. Okay, so that's the structure of DNA. RNA is the other type of molecule that's important for genetics, and it's related to DNA. It's really closely related. The key difference though is it's only got a single spiral, so we call it a single helix or single-stranded. So there's only one row of bases. It's like someone chopped the ladder in half and there's only one half of that ladder. The other key difference is instead of thymine, which DNA has, RNA has the base uracil, which just replaces it in the alphabet of RNA. Alright. The next concept is proteins. So DNA is like the instruction book. It's all the information for the cell, but it's proteins that do all the work. So this is like the worker molecules of the cell. They carry things, they signal, they send messages, they speed up chemical reactions, they make things move, like your muscles. and other parts of the body that need to move. So proteins are the workers, and remember, they're just huge molecules as well, just like DNA is. You can see here, this is a hemoglobin protein. It is made up of hundreds of atoms to build up that molecule. So every protein's got a particular shape, and that helps it do its function. Just like I listed before, so there's a whole lot of different categories of proteins. You don't need to memorize any of them, but you learn more about them as you do more biology. Okay, and the key thing is, how do genes and DNA link to proteins? And this is the cookbook analogy that I always talk about. So the example using cookbooks here, if you have the whole set of cookbooks on your shelf, that is like the genome. So the genome is all of your genetic information that's contained in one cell. Every cell has these set of cookbooks. They're exactly the same. And it's all the instructions that any cell would need to function. And then if you look at just one cookbook, well, that's like one chromosome. So remember, a chromosome is a single strand of DNA, all wrapped up together. And so that contains some of the instructions because it's only one book in the set. And then if you look inside that book, you've got a whole lot of words and recipes because it's a cookbook. And that's the DNA molecule itself. And then if you look at a particular section of the book, you'll find a recipe. And that's the instructions for making a cake. And in the chromosome, what you're looking at is a section of DNA, like you can see down here. And that section of DNA we call a gene. It's a particular set of bases that tells a cell how to make one protein. And so therefore, just like you make a cake by following the recipe, the cell makes a protein by following the recipe on the gene. And that process happens through protein synthesis, which we'll cover in a second. Alright, so we just looked at chromosomes before. Here's some more detail about how they actually package up together in the cell. Because remember, you've got multiple chromosomes in every cell, and they're such long and thin molecules, they're like strands of cotton. The cell doesn't want to just have them all jumbled up together because they'll actually tie up in knots and they'll be all disorganized. So chromosomes are arranged and packaged neatly so that they don't get tangled and they're easier to manage. And remember, the key word for how DNA is organized in chromosomes is supercoiling. So the DNA coils up. It actually coils around these round protein molecules. They're called histone proteins. So it looks like beads on a string, basically. And then those coiled up DNA and proteins, they start to coil, and then the coils start to coil. So there's layers and layers of supercoiling until we get to the fully packaged chromosome. And we call that material being made of chromatin. So chromatin is just the DNA and the proteins all packaged together. Alright, well, how do scientists actually look at chromosomes? Well, they do this process called karyotyping. We've all seen these before. This is a karyogram. So once the scientist has done karyotyping, that means they've stained the chromosomes inside one cell, and then they've taken a picture of it, a digital picture, and then they've edited the picture so that all the chromosomes are paired up in their pairs from biggest to smallest. And we can clearly see here that this is the karyogram of a human because we've got 23 pairs of chromosomes. Their full name, the proper name is homologous pairs. So obviously 23 pairs means we've got 46 chromosomes in total in every one of our cells, except for our sex cells. All right. And remember, there is a set of special chromosomes, and these determine the sex of the person. In humans, anyway. It's different for each species. So these are called the sex chromosomes. We've got X and Y. And remember, someone who is biologically female, they'll have 2 X chromosomes. And someone who's biologically male will have an X and a Y. Of course, that's just talking about the sex of someone. We're not talking about gender at all here. And we're not talking about intersex people because there are some other combinations. Some people have just 1 sex chromosome. Some people have 3, 4, or even more sex chromosomes. And that's some of the ways people can be intersex. All right, so we've got 2 sex chromosomes. They determine the sex. They've also got other genes on them as well. And every other chromosome we call autosomes. So they just mean all the other chromosomes. They contain all the genes. They're the cookbooks. They control all the information for making the proteins that a cell needs. Okay, next we are looking at different ways that DNA and chromosomes can go wrong and cause genetic disorders. So there were 2 big categories of genetic disorders. So something can go wrong on the chromosome level. So we call them chromosomal disorders. This is what our research cat was about back at the start of the topic. So in a chromosome disorder, something goes wrong on the big scale of a chromosome. So in this case, this person here, they've got 3 copies of chromosome 21 here. And when you have 3 copies, we call that trisomy. So this person has trisomy 21. Anyone who remembers that, these people have Down syndrome. And overall, all these disorders where you have the wrong number of chromosomes, maybe you've got an extra one like this person, maybe you've got one missing, these are all cases of aneuploidy, which just means having the wrong number of chromosomes. All right. The other type of chromosomal disorder you can have, sometimes when cells reproduce, parts of the chromosome actually swap over. So they'll break off, they'll swap onto another chromosome, and suddenly these 2 chromosomes are different to each other and they can't pair up anymore, which is important when cells reproduce. And these type of disorders are called chromosome rearrangements. Obviously, because part of the chromosome is rearranged. So they're the big scale disorders. The small scale disorders, we just call single gene mutations. So we're zooming into a chromosome, into just one gene, which might start here and end here. And in a single gene mutation, something happens to cause part of that DNA molecule to malfunction. So maybe a base gets deleted, maybe a base swaps over and another one comes in. But some sort of mutation happens. And these are caused by mutagens, which we'll look at in a second. But if something changes in the gene, in the genetic code of the gene, then that can change the protein. And so if a protein isn't working properly, that can lead to something not working in a person as well. Can cause diseases, but it can also just cause variation. So you can have genetic diseases, but you can also just have variation. Like this is how people have different heights, different eye colors, different hair color, any types of variation are just caused by mutations in a gene and then changes in the protein. All right, we're going back to how proteins are actually made now. This is called protein synthesis. And there are two key parts of protein synthesis. Transcription is the first one, and then there's translation at the end. So firstly, transcription happens in the nucleus. We start off with the gene on the DNA molecule. So this is just on one chromosome. And when the cell needs to make a particular protein, it opens up that part of the DNA. So this section of the DNA will unwind. And then a copy of that gene gets made. And it gets copied onto RNA, an RNA molecule. So this is where RNA is useful. It is a messenger. And that's what the M stands for in mRNA. So basically transcription is photocopying the gene onto mRNA. And then the mRNA, it's the delivery person. So it takes that copy of the gene, takes it out of the nucleus into the cytoplasm, and it goes to a ribosome. And the ribosome is like the factory that's going to make the protein for the cell. And so that's what happens at translation. The ribosome makes the protein. And then that protein goes off to where it needs to go and it does its job. Alright, going into a little bit more detail. When we're in the ribosome here, so this is happening, all this process is in the ribosome. During translation, what the ribosome is actually doing is it's reading the genetic code on the mRNA, and it reads it 3 bases at a time. So you can think of it like the genetic code has a language where it's made up of 3-letter words. And we call these 3-letter words codons. And each codon tells the ribosome to add on a specific amino acid, and that builds the protein. So amino acids, they're the building blocks of the protein. It just adds them one at a time until the protein builds up, and we get the finished protein. That's more detail than we need to know for year 10, but it's an interesting point. Alright, now we are zooming out to how do whole cells actually divide. And remember, we learnt about the 2 main types of cell division. The first one is mitosis, or technically mitotic cell division, and the other one is meiosis, or meiotic cell division. Alright, mitosis, this is the much more common one in our bodies, but meiosis is the one that's important for reproduction and for inheritance. So meiosis is the key one to remember for the genetics topic. But mitosis, this is always happening in our bodies as we grow, as we repair our tissue, if we ever get an injury, and to replace any tissues as they die naturally. It's always happening in our skin, our bones, our organs, because cells don't live forever. Alright, then the key differences between these two. Firstly, in mitosis, the cells are only producing 2 daughter cells from the beginning. So 2 daughter cells get made from one original parent cell. And in this parent cell, it had 4 chromosomes. It had 2 red ones and 2 blue ones. And parent cells, because they're a normal body cell, we call them diploid. And remember that diploid means it has, in that cell, 2 copies of every chromosome. Just like we knew in a karyotype, when we could look at the karyogram, we've always got 2 copies of every single chromosome in most of our cells. And that is what we call diploid. So in mitosis, the parent cell is diploid and the daughter cells are diploid as well. So mitosis, it's just cloning the parent basically. And that's all they need to do when we're growing and repairing. But for meiosis, it's slightly different. So the parent cell is still diploid. The first difference is 4 daughter cells get made, because it basically does 2 rounds of replicating and splitting. And because it's split twice, there's only 1 copy of every chromosome in these daughter cells. And the name we give that is haploid. So in these cells in a human, they've only got 23 chromosomes. Whereas diploid cells in humans have 46, like we know. Okay, and then lastly, meiosis is so important, like I said, for reproduction, because these daughter cells that are getting made, these are the sperm and the eggs. So when meiosis happens, it's only happening in the testes of a male and in the ovaries of a female. And together we call these sex cells gametes. Always remember that. Gametes are sex cells and they're haploid. Really important for lots of topics to do with inheritance. Okay, now we're moving on to the topic of inheritance. So the first section is reproduction as a whole. How do organisms reproduce? So there were 2 main ways organisms have evolved to reproduce. The first one is asexual, and the other one is sexual reproduction. Asexual reproduction is the weirdest one for us because as humans we don't reproduce asexually. Lots of plants do, all bacteria do, a few animals do, and lots of other microscopic protists do as well. And asexual reproduction is basically cloning yourself. You don't need to reproduce with another member of the species. You can just produce baby versions of yourself, like this plant here, and they're exact genetic copies of you. So we're not really interested in that for the rest of the topic, because as humans, we do sexual reproduction, obviously, like most animals and most plants. And sexual reproduction, as we know, involves an egg cell and a sperm cell joining together and then growing a new organism from that. So again, we're meeting these gametes which we just met in meiosis. And thinking back to the chromosome number, okay, egg cells are gametes, so they've got 23 chromosomes. Sperm cells are gametes, so they've got 23 chromosomes. So when the 2 cells come together, we've got 46 in total. And that process when cells come together is called fertilization. And the resulting cell that gets made from fertilization, so here's our cell, the sperm has fertilized the egg, we call that a zygote. It is a fertilized egg with 46 chromosomes, and it's ready to start reproducing itself through mitosis to grow into a baby. All right. This is thinking back to some big ideas for how does DNA link to inheritance? How do the two genetics topics link to each other? So thinking right back, okay, genes, they're just sections of a chromosome, and they're the instructions for making a protein. And proteins, they're the worker molecules. They control and they do everything in our cells and our body. And we're always passing our genes to our offspring by doing sexual reproduction. Because we're making gametes, and then our gametes are combining with whoever we reproduce with to make a new child. So we're constantly passing our genes on from generation to generation, and that's how species survive and evolve over time. Okay, this is more detail about how genes are inherited. Remember, we've always got 2 copies of every chromosome, but because gametes only have 1 copy of every chromosome, we're only donating one of those chromosomes to our child. So this father might donate this one to one of their children, or they might donate the other one, this red one here, to their children. And the same with the mother. And it's the same with our sex chromosomes. So females, biological females, have 2 X's. Well, they're always going to donate an X chromosome to their children. So it's really the father's sperm cell that determines the sex of the child. So if a sperm cell is carrying an X chromosome, if it fertilizes the egg, then that child will grow into a girl. But if there's a sperm cell carrying a Y chromosome, and that fertilizes a child, then that child will grow into a boy in that case, biologically. Alright, moving back into mutations and also talking about alleles. So we mentioned mutations earlier. Remember, a mutation is just a change in the DNA sequence, a change in the genetic code. And when a mutation happens, we've made a new version of a protein. So here's protein 2 that's had a mutation happen. And then that slightly different protein is probably going to have a different function. And if we're thinking back to the cookbook analogy, it's still the same protein, but it might do something slightly different, or maybe it won't work at all. So here, it's still a chocolate cake, but it looks a little bit different, and maybe it tastes a little bit different, has different icing, and specks of chocolate on the top of this one. So a mutation causes a different sequence, which causes a different protein. And these 2 different versions of the gene we call alleles. So alleles are just different forms of a gene with a different code, slightly different code. All right. We talked about mutations, so that's a change in the DNA sequence. Well, they're caused by these things called mutagens. And there's a few different categories of them. Radiation can be a mutagen, so UV light and X-rays. That's why you need to put sunscreen on and protect your skin, because UV light causes mutations, and that can cause skin cancer and lots of other issues. And that's why you need to wear protective layers when you go and have an X-ray as well. Lots of other chemicals can cause mutations, and some bacteria and viruses can cause mutations as well. So these are all mutagens. Alright, moving further into inheritance now. This is the idea that now that we know there are different alleles of a gene, that's going to affect our characteristics now, because we always have 2 copies of every gene, because we've got 2 chromosomes, 2 copies of each chromosome. So these are the key words to focus on. It's character, genotype, and phenotype. Character is just a particular characteristic that an organism has. Like in this case, it's the fur color of a cat. That's the character. The genotype, that is just the list of what are the 2 alleles that this particular individual has. So this cat here, it's got the genotype big A, because they're the 2 alleles that this cat is carrying in its chromosome. This cat down here though, it's got the genotype small a, because it's carrying 2 small a alleles. And then the phenotype is the actual physical characteristic that's caused by the genotype. Pheno is physical. And trait is the other word for phenotype, physical trait. So in this case, we've got a black fur phenotype in this cat. And the other cat down the bottom has brown fur as its phenotype. So it's like the version of the characteristic. Alright, we also give names to the different genotypes. So if you have 2 copies of the same allele, maybe you've got big M, we call that homozygous. Obviously, homo just means the same. So 2 copies of the same. You could also have 2 small m's, that's also homozygous. If you have 2 different alleles of a gene, we call that heterozygous. Hetero just means other. Alright. And now we're getting onto patterns of inheritance. So different ways that genes can interact and cause a different trait in an actual organism. And there are 3 main patterns. The first one is called complete dominance. This is the most common and the easiest one, so you learn it first. For complete dominance, it's all about the heterozygous person here. So for complete dominance, if you're homozygous for one of the alleles, you're definitely going to have that trait. So this cat here is homozygous for big A, and they have black fur. This cat down here, they're homozygous for small a, and so they're going to have brown fur. But the key is, in a cat that's heterozygous here, only one of the traits, one of the phenotypes is actually visible. And in this case, it's black. And so that tells us, okay, the black phenotype must be dominant over the brown phenotype. So black fur is dominant over brown fur in this case. So you can think of it like the dominant trait is masking or covering up or dominating over the other trait, which we call recessive. So black fur is dominant, brown fur is recessive. And then to show that, which I've already done in this question, we use a capital A to represent the dominant allele, and we use a lowercase a, or whatever letter we're using, to represent the recessive allele. Okay, the next type of dominance is called incomplete dominance. And in incomplete dominance, you have 2 phenotypes. So in this case, we've got a red flower in a pea plant and a white flower in a pea plant. Again, if you have a heterozygous individual, that's always the key in these patterns. But in incomplete dominance, the heterozygous individual, they have a mix of the 2 phenotypes. So instead of only one phenotype showing up in a heterozygous plant, which is what happens for complete dominance, there's neither of them are dominant. So they both show up at the same time and they get mixed together. So we say red flowers and white flowers are incompletely dominant, and so this plant here, because it's heterozygous, it's going to have a mixture of the two. So and this is an example of the way we write the alleles for incomplete dominance. You can use a superscript, which just means a small letter at the top. So in this case, we've got C as just the gene, and then you've got R to represent the red allele, and then a W to represent the white. So this plant is homozygous, this plant is homozygous for the white, and this plant is heterozygous. You can just use 2 capital letters as well if you want, particularly at year 10. So that would be completely fine as well. Alright, the third pattern of inheritance is called codominance. And similar to incomplete dominance, in this case, if you've got a heterozygous person, or a heterozygous cow in this case, so here we've got an R and a W allele. In this case, instead of the 2 phenotypes being mixed together, you get patches of the different phenotypes across the organism. Patches of the 2 phenotypes. And in this one, we've got the white phenotype and the red phenotype, and so when they breed together and have a heterozygous calf, that calf we call roan because it's got patches of white and patches of red fur. Codominance is all about patches. Incomplete dominance is they mix together. Okay, onto Punnett squares now. So Punnett squares are one of the main tools we use to solve problems with genetics because they can help us to predict what are the possible traits that some children are going to have. And to draw a Punnett square, we have the 2 parents, here and here, and we write down the 2 possible alleles for this gene that they might donate in their gametes. Because remember, gametes are haploid. Each gamete only has 1 allele of each gene. So for this pea plant here that's purple, it could either donate a big B allele or it could donate a small b allele. And it's the same for this other parent plant over here. It could donate a big B allele or it could donate a small b allele. And so then we're basically talking about probabilities now. So if this gamete happens to fertilize this gamete, then the baby pea plant will have big B as its genotype. If this B small b allele happens to fertilize the big B allele here, this pea plant will be big B little b. The same here. And then if the 2 small b gametes come together and fertilize, then we're going to have a small b flower. So that's the genotypes, and then you can look at the phenotype as well. And it looks like in this case, purple flowers is the dominant trait. So all these heterozygous babies will have purple flowers. This one's definitely purple. And the only one with white flowers is if you're homozygous for the recessive allele. All right. Then you need to be able to solve problems using Punnett squares. This is the main area to do with inheritance. So here's an example. Let's work through it. We've got a trait to do with dwarfism, and it gives us the alleles. So since we've got big capital D for dwarfism, we know that dwarfism is dominant already. That's what I'm thinking. Okay, and we know that the normal trait must be recessive. So the question goes, a hybrid dwarf and a normal height person have a baby. Well, hybrid is just another name for heterozygous. So we know we've got a heterozygous person who is dwarf and a normal height person, and they're having a baby. So let's put the heterozygous person at the top. They must have a big D allele and a small d. And a normal height person, well, normal is recessive, so they must have 2 recessive alleles to be normal height. And then we fill out the Punnett Square. So big D and small d are coming together here, big D and small d again, and then 2 small d's here, and then these 2 gametes are coming together here. So the possible genotypes that could be made, well, we can either have a big D small d or small d small d. The possible phenotypes, well, dwarfism is dominant, so this genotype is going to be a dwarf person, and this genotype, they're going to be a normal height person. And then questions often ask about the ratio or the percentage. So to work out the genotype ratio or the genotypic ratio, we need to just work out, using our math skills, what are the percentages of each of these genotypes? Well, we can see here it's 2 out of 4 are going to have big D, small d, and 2 out of 4, so a half, are going to have small d, small d. So we can say it's always good to work in percentages first. 50% heterozygous, 50% small d, or homozygous. And then you can also write that as a ratio. So 50 to 50 is the ratio, but using our math skills, we know that 50 to 50 simplifies to 1 to 1, just like simplifying a fraction. And we think about the phenotypes as well. And in this case, it is 50-50 again. So that's pretty easy. 50% of the children in this family will be dwarf, on average, and 50% will be normal height. And again, that's a 1 to 1 ratio. And then we've got another question at the bottom here. What are the chances the child will be of normal height? And that is 50%. Just like we worked out. Okay, next question. So here we're talking about mice, and these mice have made 50 offspring, and all of them are hybrid or heterozygous. And we know we're talking about an albino trait now. Remember, albinism is you can't make coloured pigment in your skin or your hair. So albino people have completely white hair, completely white skin, because they can't make any coloured pigment. And we can see here, because albino is the small a, albino is recessive in this trait, for this gene. Okay, so in these mice, 50 offspring were made and all of them are hybrid, and no albino mice were born. So what can we conclude about the parent's genotype? And we have to prove it using a Punnett Square. Okay, well, I'd be thinking, okay, let's set out our Punnett Square, and we know that all the offspring were hybrid or heterozygous. So we're working backwards in this one. We know all the offspring are heterozygous, so they must be big A, small a. And that means a big A must have come from one of the parents. And that means, in this case, a small a must have come from the other parent. And then again here, a big A must have come from one of the parents, and the small a must have come from the other parent. And you could play around with it, you could do some trial and error, but I can already see here that this is the only way that we're going to get every single baby mouse being hybrid or heterozygous. So what can we conclude about the parent's genotype? Well, one of them must be homozygous dominant, and the other one must be homozygous recessive. So these are the parent's genotype. The parent's genotype. Okay, next question. We've got, here are some chooks that are called Andalusian fowls. We've got black individuals and white individuals are homozygous. Okay, so since we're looking at two different letters, straight away I'm thinking, okay, this is either incomplete dominance or codominance. And then it says, when a black bird is crossed with a white bird, the offspring are bluish grey. And when black mixes with white, that's like they're blending together to make grey. So now I'm thinking, okay, we are looking at incomplete dominance for this question. And now it's asking, what are the probabilities of each genotype and phenotype if a black individual, which must be BB, is crossed with a bluish grey? So they must be heterozygous. Let's prove it in a Punnett Square. So a black individual has to donate a B or a B, and a grey chook is going to donate a B or a W. And then we'll fill out our Punnett Square for the possible baby chicks that could be made here. There we go. And we can see it's going to be 50-50. 50% of the chickens are going to be black and 50% are grey. 50% BB black, 50% BW and grey chickens. And the last question is, two bluish grey chickens were crossed. What are the possible phenotypes and genotypes of their offspring? And give the probabilities. Okay, so again we need to set up a Punnett Square to prove it. But we've got two bluish grey which are heterozygous this time. So both of them are heterozygous. And in this case, we're going to have three possible genotypes and three phenotypes. Okay, so the first one, a quarter or 25% of the chickens are going to be BB, which is black. These two here, they're heterozygous, so that's 50% because it's 2 out of 4. Will be BW, and they are going to be grey chickens. And then 25% will be white, which is WW. Okay, so that's an example of solving a problem that has incomplete dominance. So you're still using the same method, but you need to recognize which type of pattern of inheritance it is. And all that information is going to be in the question. Alrighty. The last section in inheritance is pedigree charts, what they are, how you draw them, and how you read them as well. So remember, pedigree charts, they're just a fancy version of a family tree, and they're really useful for tracking the inheritance of a particular trait or a particular disease through the generations. And that's what we're showing in each row of a pedigree chart. So the 1, 2, and 3 are the different generations. So we've got grandparents, children, and grandchildren. The lines across, the horizontal lines, they're called marriage lines. Bit of an outdated phrase, but it just means these two people had children. They don't necessarily have to be married. We also have horizontal lines like here and here, and these are called sibling lines because they're showing these siblings are related to each other, that they're brothers and sisters. And we also have, sometimes, but not always, we have individuals numbered as well. And if they're not numbered, you can just do it in your head or you can write it out like I'm doing now. 1, 2, 3. Okay. And to recognize an individual and to actually name them, we write down their generation first and then the number that they are. So this is, to identify this person, we're going to call them individual 3, 1. Because they're in the third generation and they're the first child. This person here, we would call 2, 2. And so on. This person here is 3, 7. And you can name anybody in the pedigree that way. Alright, the last thing to do to recognize or to read and to draw pedigree charts is to know the different shapes and what they mean. So first of all, we have squares. Squares mean males, biologically. Circles mean biological females. If you have a filled in square or circle, that means you have that trait or you have that disease. So we often call that affected. And then if you have an empty circle or square, you're not affected by whatever trait or disease we're talking about. So let's look back at these individuals here. So individual 2, 2, she is a circle, so she's a she, biologically, and she is not affected by this trait. So she's an unaffected female. Individual 3, 1, she is affected. She's a female as well, whatever trait we're talking about. And individual 3, 7 is a male, and he is not affected in this case. So you can see here in this family, the grandfather had the trait, and then his son and one of his daughters and another one of his daughters had the trait, and then the son's daughter had the trait, and then this daughter had 2 children with the trait, a daughter and a son, and 2 of her other children didn't have the trait. Okay, that has covered in moderate detail all the concepts and most of the vocabulary for the genetics topics. So there's a lot there. The key things to focus on though are pedigree charts, being able to solve problems. That's the main skill we're testing. Alright, good luck with your revision and good luck with your assessments.